Hello, and how are you? Welcome to today's session on cell membrane specialization. My name is Winnie Bajawa. If you're new here, this is a channel that focuses on content, lessons, and interviews relating to healthcare. And if this is your area of interest, kindly remember or kindly subscribe. And if you're a returning subscriber, we really appreciate you coming back and being with us. So be with us throughout the class. And in this session, our main focus is going to look at the cell membrane and features that actually make it efficiently work or make it unique or enhances the ability to do their work better. And we will look at two things, that is the microvilli and the cell junctions. So let's delve in and see what is in for us. Microvilli, these are finger-like projections and they're found on the surface of the cell membrane. Their main reason is to increase surface area for absorption. They are found commonly in areas that do processes of exchange of materials and could be in areas like the respiratory epitheliums, can be in the digestive system covering. And here are our microvilli. I hope you can see them on the diagram. So the microvilli, what they do, because of the nature of projections, they actually increase surface area, and this surface area are key for the purpose of exchange of substances or for the purpose of movement of molecules and solutions across. So we will discuss them though in more details. And then the other part that we're going to look at is the cell junctions or the membrane junctions. And junctions by definitions are connections or areas of contact between cell and another cell. So they are areas that will allow us to join one cell to the other. And so there are three main junctions we are going to look at, and that is the tight junction, which is known to be very impermeable. We have the desmosomes, which are the anchoring junctions or the adhering junctions, and the gap junctions, whose main role is to ensure communications. So if you look at the cell here that I'm using to display, we have the areas where one cell here is connecting to another cell on this side. So for the cell to connect to each other, we need areas of contact, or we need now what we are calling junctions. So we have this here is a junction, there's another type of a junction here, and then we have another type of a junction also here. The space in between the two cells is what you're calling the extracellular space. And so with these different types of junctions, they all have different characteristics. The tightness is not the same. The functions are also not the same. But now we have the microvilli here. In the microvilli, we've said these are finger-like projections, which are minute. And because they're finger-like projections, they are extensions from the plasma membrane that project freely or are exposed from the cell surfaces. So number one, they increase the plasma membrane surface area tremendously and are most often found on the surface of absorptive cells, e.g. in the intestines or in the kidney tubule cells. So what these areas do because of the microvilli is, we like it say, they increase the plasma membranes. And because they increase the surface area of this plasma membrane, they allow a larger surface for exchange of material, be it by the different mechanism of cell transportation, and this allows more efficiency for the cell membrane. So here is our cell. So this is the plasma membrane here, and sorry, these are the cell itself with the content and the organelles in it. And on each cell, like we say, they need areas of contact or connection, but on the surfaces, we get to have our microvilli. So with these microvilli, all these are expansion of the cell membrane, or these are extension of the cell membrane. And with this, it's able to increase the surface area for more exchange of substances, for more transportation of substances across it. And that's basically in summary of what the microvilli is. It's a minute finger-like extension or projection from the plasma membrane that projects exposedly to the other part of the cell, and the main purpose is to increase surface area for exchange of materials, or to increase plasma membrane surface area for exchange of materials. Commonly found in absorptive cell surfaces, e.g. in the intestines or in the kidneys. We can also find microvilli in the respiratory systems that allows it to gaseous exchange. And the other part are the cell junctions, and we've said cell junctions are points of contact of one cell to another. And the cells are connecting to one another, or they create contact with each other using different types of junctions. But before we go into mentioning them, I want to explain more on how this contact happens. So here we have several cells. These are 4 cells here. And we have our microvilli, the projections from the cell membranes. But again, we have one cell connecting to another cell. So this area of contact here is what we are calling the junctions. And so the junctions occur of different types. So with these junctions being of different types, it means their tightness will also vary, or their functions will also be different. So we will discuss 3 main junctions, or 3 main types of junctions, and we have the tight junction. So with the tight junction, we are told this is a very, very tight connection between 2 cells. Doesn't allow materials to cross through, and it's mainly impermeable when it comes to passage of materials across. Then we have the second type of junction that is known as gap junction. So the gap junction, just like the name says, gap, it allows spaces in between the cells, and it also allows materials to cross. And they have a special connection between them known as the connexons. So this is gap junction. And then we have the desmosomes. Desmosomes are known for adherence. They're mainly in place to adhere the cell to one another, and they also have special characteristic which is made of materials we call plaques. And with plaques, they also have adherins, and the catherines we have the intermediate filaments. So these are features that make the desmosomes unique, but the main purpose is to adhere cells to one another. And the other junction that we'll check on is the adhering junction, although this will not be our main focus in this class, because mostly, or most books, when you read on the desmosomes, they also call them the adhering junctions. And if you even look at the features, they literally have features that are similar to the desmosomes characteristics. So you can see they have cadherins, they have the filaments, and all these are similar characteristics to the desmosomes. And the cell junctions, by definitions, these are complexes that provide contact between cell and another cell. They're specialized areas, and the main reason is to provide that contact between one cell to the other. So we have the 3 junctions that we are going to focus on, and that is the desmosomes, that we also call the adherins. We have the tight junctions, and then we have the gap junctions, also known as the nexus. So let's start with the first junction, and that is the desmosomes. So with the desmosomes, we are told they're commonly found between epithelial cells. Yeah. And the desmosomes, because we have space in between the cell, what we are calling the intracellular space, or gap, it's about 25 nanometer wide, and what it does is the desmosomes, or this space rather, contains adhesive glycoproteins that we call desmocollins. So in between this intracellular gap or space, we have the desmocollins on it, and then we have the intracytoplasmic densities known as the plaques that are present beneath the plasma membranes of both junctions. Now these plaques are very key because they provide supporting base for the junction. So groups of intermediate filaments are also inserted into these plaques to make hair loops that are very important in strengthening the junction. Remember, this is a contact point between 2 cells. So let me describe for us what is unique about the desmosomes. So we are mentioning the plaques, and the plaques are key base support. So what they do, there's one plaque underneath the plasma membrane of one cell, and another plaque on the other plasma membrane base of the other cell. So what these plaques do, they create the base, or the points of adhesions for these two cells. And as they pull together, we have the filaments, or the connecting filaments. So these interconnecting filaments, they're the ones that pull our cells together to create a connections or to create the adhesions. But again, in between the intracellular space or the intercellular space, we have our desmocollins, and these are glycoproteins that create more adhesions between the cells. And then to give it more support, we have the cytoskeleton filaments. So these are filaments that give the hair loop-like appearance, and they also provide the connecting agents for our desmosome or our junction. So you need to appreciate that the desmosome, the main reason is to adhere two cells or to adhere one cell to another. And for this reason, you really need to make it strong or to support it to give that adherence purpose. They are not that tight. They have a bit of loose because we have a space in between them that is around 25 nanometer. But again, if you really don't remember what about the desmosomes, just remember the characteristics it has. It has connecting filaments, it has cytoplasmic plaques, it has cytoplasmic filaments, and there's an intracellular space of around 25 nanometer wide. So that's a basic remembrance to have about the desmosomes. Second type of a junction is the tight junction. And just like the way the name describes it, is that this junction is very tight. The cells are very close to one another and they're apparently fused, leaving no intercellular gap or no intercellular space. So for the desmosomes, remember it has an intercellular space of around 25 nanometer in terms of width. But for the tight junction, we do not have this. And the main purpose of tight junction is to create the contact or to create the connection between one cell to another. But unfortunately, it does not allow materials to pass across this particular junction. And so as you can see with all these tight junctions here, they are all connecting directly or tightly and does not allow any material to cross here. Yes, we have the space, but this space is not as wide as you may want to describe it. So we have our cell A here and cell B, and they're connecting at the tight junction very tightly together, contacted into each other, fused, and there's no gap between these two cells. So in short, you cannot expect materials to cross or materials to move from one cell to the other in the cases where we have tight junctions. And the last cell junction is the gap junction that you are also calling the nexus. So with the nexus, as the name says, gap, what this junction has is that it allows materials to cross. So we have the membranes of the cell are there to each other or rather connect to each other, but there's a gap with this type of junction that is around 2 to 5 nanometer that will permit materials to cross. So the gap junctions are said to be widely distributed in the body, and you can find them in very many places, e.g. in the cardiac muscles, in the smooth muscles, the liver, the kidneys, and also the thyroid glands and skin. Main reason is this allows materials to cross. So in the smooth and the cardiac muscles, we are also told the gap junction allows spread of impulses from one cell to the other. So just to remind us that in the smooth and cardiac muscles, during the muscle uh, contraction and relaxation, we have the nerval stimulation to facilitate that. And for the purpose of passing one impulse to the other, then the gap junctions allows that to happen. So the gap junction literally provide communication between the adjacent cells, and their intracellular gap is traversed by a hollow tube structure. So let's make all this word into a picture and understand how the gap junctions looks like. So we have two cells. We have cell 1 and cell 2, and these are their cell membranes or their plasma membranes. They are connecting together. But now we are told in this space or in these two cells, there's space in between, and we are calling the intercellular space. So our intercellular space is about 2 to 3 nanometers and permits materials. But now the materials on the gap junction do not really permit through the intercellular space. It permits through the channels. And this is what you are calling the hollow structures. So these hollow-like tubes, what you are calling the hydrophilic channels. Hydrophilic means They are water loving, or meaning they will not react with water in case they're put in aqueous solutions or they're in contact with the cytosols. So they'll allow materials to cross from one cell to the other. And these points of gap or these points of connection is what you're calling the connections. So if you forget anything about gap junction, you can just remember the connections and the fact that it allows materials to cross from one point to another. And we've also said it's found commonly in areas that have connections, e.g. in cardiac muscles, smooth muscles, liver, kidneys, thyroid, and the skins. And the gap junctions creates allowance for impulses to cross or to travel from one cell to another. So literally the gap junction is the one that has the most function when it comes to allowing materials to cross. And this is unlike the desmosomes, unlike the tight junctions. And so if this is your area of interest, healthcare and any other related context to health, please remember to comment what did you like about this video. Also share with your friends so that they can watch more on our work. Subscribe, and above it all, please give it a thumbs up. Thank you so much for being with me to the end. My name is Winnie Barawa. Bye bye, and see you in our next lessons.